Okay, thank you. So the way I play it, unfortunately, I can't see anybody, so please interrupt if I'm not being clear. So thank you very much for the invite, and I'm very sorry that I can't be there in person. But today I want to talk about glucose transport in budding yeast. And as you all know, glucose is the world's most abundant sugar, and it's the preferred carbon source for many microbes, with the pathways that metabolize sugar being very highly conserved bacteria to our own cells. So we sort of became interested in glucose in yeast because yeast, like many microbes, prioritizes glucose. But what was surprising to us is it has 18 potential glucose transporters. So the ones that people have studied the most, and the ones that at least we know yeast need to grow aerobically on glucose, are the ones I've marked in blue here. So there's sort of 7 of them. 2 of them have evolved to become glucose sensors, which are these 2 green ones here, which I'll return to later. But it's just surprising that there's so many, at least for us. So there's 7 of these transporters. And so the first question we started to think about is, well, why not just have one which has got a high affinity for glucose? Why have this large number that I mentioned before? So the way that these transporters all work is that they use facilitated diffusion. So that means that the protein is the transporter is sitting in the membrane, and then thermally fluctuates back and forth between facing the extracellular space and facing the intercellular space. And it does this without consuming energy, it's thermally. And so the transport is simply down gradients. So typically there would be more glucose outside the cell than inside, because inside the cell yeast phosphorylate the glucose, which prevents it from binding the transporter. And then you can think of then if this transporter is providing a flux, which we can describe with a Michaelis-Menten type equation, but where the substrate is this gradient, or this difference in glucose concentrations between the inside and the outside. Okay, and others have speculated that this kind of transport is potentially a rate affinity trade-off. So that this was particularly in this paper here, they first came up with this idea. And so what do we mean by that? So the rate is sort of obviously this k-cat describing flux, and the affinity is given by 1 over the km. So something with a high affinity has a low km. So why might there be a trade-off? Well, we can try and think about what's happening as this transport takes place, and I can do that with one of these free energy diagrams. So energy is plotted here, and the extent of the reaction is going along the x-axis. And so initially we have glucose free and the transporter sitting in the membrane, but not bound by glucose. Glucose binds, and this gives a lowers the free energy, shown by the purple line, if we just focus on that one. So now we're bound, and now the transporter has to flip across to face the other side, the inside of the cell. Let's assume that there's a free energy barrier to that. So this is sort of the rate limiting step, if you like, in the process. And then once it's flipped across, then glucose has to unbind again and then be released so that it can be metabolized. And so as I mentioned, the affinity then is given by the depth, by the height of the depth of this well in the free energy. So the lower the well, the higher the free energy, and the higher the affinity. And the rate is given by the rate of crossing this barrier. So it's given by 1 over the height of the barrier. So then you can imagine that if I then have a different transporter which has a higher affinity, to have a higher affinity, I have to make this well deeper. So I lower this well. By doing so, then I've made this activation barrier higher, and so I necessarily decrease the rate. So that's sort of the intuition about what's going on. So I hope that was clear. So that I have one free energy well determining the affinity, and when I change this one, I necessarily increase the rate. So you can do that. So if I pick if I make a mathematical model, and then I pick some parameters and calculate randomly and calculate the rate and affinity, you can see quite clearly that there's a relationship between the two, and it's of the form of a trade-off. So if the rate is high, the affinity must be low, and similarly, if the affinity is high, the rate becomes low. So we can calculate, as I said, this model is straightforward, so we can go ahead and calculate everything. If, in particular, if I assume that this rate of flipping back and forth across the membrane is identical whether or not glucose is bound, in other words, that these R primes are the same as the R's, then the expressions for the kcat and km are a bit simpler. Notice that these are kind of effective kcats and kms, where this is the effective substrate, the difference in glucose concentrations. And that internal glucose inhibits the transport. Okay, so you can see there's a GI for internal glucose here and a GI here. I guess I should explain the other parameters. So R is, as we said, is the rate of flipping. And the other important one is this KD, which is given by the ratio of these two rates. And so it's the dissociation constant of glucose binding to the transporter. So once we've calculated these kcat and km, then we can vary the parameters. We could vary R, this Fi, or the KD, to see how kcat and km vary. And you can show that there is a trade-off, so that if I increase kcat by varying one of these parameters, then I necessarily decrease km. But only two of those parameters turn out to be important. So that's these Fe and Be, which are the ones describing how glucose binds outside the cell. So if I vary the other ones, for example, R, then I get a trade-off. But when I've placed how these two parameters are changing into my equation for the flux, if I increase R, the flux always increases. So even though kcat is increasing and km is km, the increase in kcat dominates the change in km, so the overall flux increases. So changing R is an interesting parameter in the sense that we might imagine the cell optimizes this because it doesn't suffer any loss for increasing R. But if I change these two controlling the binding of extracellular glucose, then there's a possibility that I can actually reduce the flux because of the trade-off. So I'll show that here. So what that means is that for each particular concentration of extracellular glucose, there's a transporter that works best. So if I can characterize the transporter by its Km, and remember if I know Km, then I can predict what Kcat can be approximately because there's this relationship between the two. And normally, so what I'm plotting here is the flux through the transporter versus the value of the Km. And you can see in the equation, if I increase Km, then I expect the flux to fall, which is what you can see is happening here. So the larger Km, this gets bigger and my flux decreases. Because, however, we have a relationship between Km and Kcat, by increasing Km, I also increase Kcat. So in this region here, when I increase Km, my flux doesn't fall because the corresponding increase in Kcat dominates. So I get an increase in flux until eventually Kcat becomes saturated. And then as I increase Km further, then it falls again. And so what this means is that for this particular concentration of glucose, which was 10 millimolar in my simulations, then the optimum Km, it's given by here, which is close to 10 millimolar too. And this is the transporter I should have if my extracellular glucose was 10 millimolars. If I change extracellular glucose to 1 millimolar, then the same argument holds, but now the transporter I should use, if I want to optimize flux, should have a Km, whatever, of about 5 or something millimolar, and so on. So because of this trade-off, each transporter, if we characterize this by its Km, has a range of glucose concentrations where it transports better than any other transporter. So what we then did is we went to the measured values of these Kms for the hexose transporters, the glucose transporters in yeast, which is sort of given here. And then for each of them, we can estimate what the flux would be. And so I plotted that here, and I've for the reasons that will become clear in a second, I've inverted the axis. So here is high glucose, and here is small glucose. And so if I look to read this plot off, you can see that the flux of this transporter, the pink one, is best when glucose is high. Okay, the pink one is this one, which has got a high Km. Okay, so in this region of glucose here, this one is best. Then the next one is this one, the purple one becomes best for this region of glucose, which is shown here. Then this one becomes best, and so on. Okay, so because of their Kms, we can sort of predict the order in which these should be expressed if the cell wants to optimize the import of glucose. Okay, so for high glucose, it should be this one, and then this one, and then this one, and so on. So to see if that prediction of the order is correct, what we did is we used a fluorescent protein to tag the hxts in yeast, and then we put these cells into 2% glucose, which is about 110 millimolar, and let the cells consume the glucose and watch what happened over time. So the dotted line first is the optical density, so the number of cells that we have. And so over time, the cells consume the glucose and this increases. And in fact, you can think of this as some measure of the amount of glucose that's available in the media. So the higher the OD is, the less glucose is going to be present because the cells will have consumed it. On the y-axis, no, the other y-axis, I'm plotting the fluorescence per cell. So that's, you can sort of, and then we have time here. And you can see that the different hxts come on at different times when the concentration of glucose is different. And they peak particularly at different times. And the times at which they peak is consistent, at least for most of them, with what we see. So the purple one here, hxt1, peaks first, is what we see. Then the next one is the one with a slightly lower Km, then peaks, and then this one, and then these two. So the order is roughly correct. Of course, it's biology, so it's not perfectly correct. So there's two transporters that seem to do something different, which are hxt5 and hxt2, for those who are interested in it. But generally, the order seems to be correct. And this is sort of consistent with these transporters necessarily having a rate affinity trade-off because they use facilitated diffusion. So which means that each of them will transport best in a certain range of glucose concentrations. And I should also point out there's other explanations which would work in parallel with what we're saying from Bast-Toysing's group, which are published a few years ago. Okay, so that's what we expect, and what we understand because of this rate affinity trade-off. And so the way that you can imagine, or the way that we imagine these transporters evolved is that we know that the first transporter was most closely related to this gray one, hxt5, and then it evolved through gene duplication. And so what we might expect if there's a rate affinity trade-off is you have your first transporter, it evolves, so it has a particular value of binding, rate constants of binding to glucose. And so it's a particular range of glucose concentrations over which it's best. When it gets duplicated, then we might expect these parameters to evolve so that the new transporter is optimum in a different range of glucose concentrations, so that the cell overall can do better in a wider range of glucose. And that is sort of consistent with what we see if we look at the evolution of what happened with these. So as I said, hxt5 in budding yeast seems to be most closely related to the original transporter. And then Kevin Correa in my lab did this analysis to show that it was then duplicated, which gave rise to hxt6 and 7, which are the ones with low affinity. So hxt5 itself currently has medium affinity. So then we get this low affinity ones. The next events were duplication of 2 and 4, which have medium affinity. So again, we're extending the range of affinity. And more recently, we have had further duplications to create the last two, 1 and 3, which have low affinity and have extended the range of affinity even further. So this is sort of consistent, at least, with how these have evolved. Although, of course, the affinities that I'm describing are affinities for the present day yeast, which may have nothing to do with the ones in the past. But anyway, the duplications seem consistent with the idea that you have one transporter which is best in a certain region, and then you duplicate this, let parameters evolve so you can explore a new region, and then do this again and again to gradually tile, if you like, your optimal transport over the range of glucose concentrations that are present. Okay, so that's how we think, the reason why we think there's so many of these transporters. But then the cell has the problem is it needs to match the particular transporter that's optimum to the current concentration of glucose. So how does it do that? How does it solve that problem? So this network has been studied a lot. So I'll just describe how it works. So there are two main pathways. The one that's been most of the attention has been on is this SNF3 RGT2 pathway. So these are two glucose sensors. So I mentioned at the beginning that people think they used to be transporters and then lost the ability to transport, but work similarly so that they flip back and forth between the inside and outside of the cell and reveal the amount of glucose that's present extracellularly. And these two sensors control the activity of two repressors. The way they do that, I'll come to a bit later, but it's not too important. But you can think of them both as repressing the activity. And then these repressors then go and control level of the transporters, which are the hxts. And there's another pathway that responds not to extracellular glucose, but to the effects of metabolized glucose. It's not exactly clear what it responds to. It's possible it's levels of the ADP. But anyway, we can think of it as responding to metabolized glucose. And it also, this kinase, which is AMP kinase in mammalian cells, it controls another repressor, MIG1, inhibits it, which also inhibits the hxt gene. And then finally, there's another repressor, MIG2, which is a paralog of MIG1, and that is actually controlled by the SNF3 RGT2 network. Right, so there's a lot of repressors controlled, but I'll come back to it in a second. But at the moment, what we're going to focus on is these two sensors and these initial two repressors which directly repress the hxt gene. Okay, so that's what I said. We'll focus on that, and we'll ignore this pathway for the moment. And the way that we decided to examine what's going on with these is to perform deletion studies. So we'll delete each of these components in turn, and then see how the expression of the hxt changes. So the type of experiments that we have are shown here. So let me try and guide you. So I've got fluorescence per OD. So these are these batch culture experiments where we have the cells in a test tube, we add a load of glucose, and we watch what happens over time. Okay, over about 20 hours. Fluorescence per OD. The black line is the wild type. So, sorry, the colors aren't so good, but that's this one. And then the colored lines are the different mutations where we've deleted components of the sensing network. And in some cases, this causes higher expression, in other cases, much lower expression. We do that in two different concentrations of glucose. And then having it as a function of time isn't so useful because the network is about sensing glucose concentrations. You can measure glucose using enzymatic assays, but that's difficult to do for many experiments. So instead, the way that we try and estimate what the glucose is we look at the corresponding OD, optical density of these cultures. Okay, so I should have it on here, but I don't, but this is something that goes up and then plateaus as the cells use up the glucose. And so we can convert that optical density into an estimate of the glucose concentration. So that's what I've sort of shown here. So it's a very simple estimate. So the glucose content, so it's at a maximum at the initial concentration of glucose when the OD is at its minimum value at the start of the experiment over here. And glucose reaches its minimum concentration when the OD is maximum, okay, when it plateaus over here. So then this would be 0. Okay, so we just do an interpolation between these two extremes, assuming a linear relationship. But this allows us then to convert the time axis into a glucose axis, or an approximate glucose axis, where I've again reversed it. So we start with high glucose corresponding to initial time, and then decreases over time as the cell consumes it. And that just helps emphasize the important parts. Okay, so we generated this data. We then, so this is a picture of Sherlock Holmes, of a statue of Sherlock Holmes. Arthur Conan Doyle, as you know, who wrote the books, lived in Edinburgh. And this is a quote, because what we did initially is we did lots of what we thought was clever analysis to try and compress this data into a single figure where we compare growth to wild type, and we looked at Jacquard indexes and did lots of different things. But retrospectively, what we really should have done is just look at the data that we had, try to understand it just from the time series. So there's a lot. I was surprised actually how much information you can get from just looking at these and thinking about what the network is without any sort of additional analysis. So picking one of the transporters, the hxt1 here, so you can see the wild type is the dark curve, which is buried in these colors somewhere. Okay, again, glucose starts high and goes to 0 during the course of the experiment. And very strikingly, if we delete STD1, that's one of the repressors, we get very high expression. But if we delete MTH1, the other repressor, the curve is the same as wild type. Then we can look at the sensors. If we delete the SNP3 sensor, the curve is in here somewhere. But if we delete the RGT2 sensor, then we get almost no expression. So the combination of these two things tells us that this RGT2 must be controlling the STD1 repressor. Okay, because we know that STD1 is the only one that seems to be affecting levels of the transporter. And we know that if we don't have RGT2, we get no expression at all. So RGT2 must be making this link to switch off STD1 to allow the wild type expression. So we have some idea of how this network, beginning to see how this network works. If we pick a different hxt, which is a different affinity for glucose, it's not quite as clean, but there's a similar effect here, where now if I delete STD1, I get a response similar to the wild type. But if I delete the other repressor, then I get much higher expression. And if I delete this sensor, nothing happens. But if I delete the other sensor now, I get much lower expression. So now I can see that this sensor here seems to be predominantly controlling this one. We can even start to explore what's happening in this part of the path of the network, which is the one we weren't initially focusing on, because of some of the feedbacks that are, of the control that's present in it. So you can see that we just said that RGT2 tends to control STD1. Okay, so I have this relationship. But then from the diagram, I can see STD1 promotes this SNP1, the AMP kinase, which phosphorylates MIG1. Sorry, which phosphorylates, so inhibits MIG1. So I have this negative relationship here. So STD1 represses MIG1. And I also have another negative relationship because STD1 actively represses the MIG2 gene. So STD1 also represses MIG2, so it represses both of them. So then if we look in our data, we see this odd behavior. So again, here's the wild type. It's the black line, it goes up and peaks around this concentration of glucose. But if we delete STD1, which is a repressor, then we get much less expression than the wild type. And this we started to understand because STD1, by deleting it, we're reducing this inhibition of these two repressors, and it's these two repressors are the ones that are becoming overactive and bringing the levels much below wild type. Now, if that was true, then if we delete RGT2, we should reduce that and we should sort of increase the levels of active STD1 and make this even harder, and make this repression even stronger and see higher expression, which is also what we see. Okay, so if I delete RGT2, then I have lots of STD1, and I repress entirely the action of these MIGs, and I should get much higher expression than wild type, which is also what we see. So if we have this bracketing behavior for these two deletions, then we know that this promoter must be repressed by MIG1 and MIG2 predominantly. So that's true for this hxt4. And if we look at some of the other hxts, we can see that's true there. So this is the one, what we see in general is that the medium and high affinity ones are controlled by these three repressors. MTH1, I showed this to you already, is where if I delete this, I get much higher expression, but not if I delete STD1. Here's the one with the map where you have the two bracketing it, which shows it must be controlled by MIG1 and MIG2. This is a high affinity hxt, and it has a similar, not as dramatic, but similar behavior. You can see that the orange line is above and the STD1 line is below. And here also, in very low glucose, these are the high affinity ones, which is where they should be expressed. You can see if I delete MTH1, I get much higher expression. And similar behavior for the SNP3. So this sort of implies that the medium and high affinities are controlled by these three repressors. And finally, the low affinity ones are controlled by this other one, STD1. Okay? If I delete that, both of them, hxt1 and 3, the levels increase dramatically compared to wild type. So from these deletion experiments, we get some idea of how the network is working. And there's this division where SNP3 controls MTH1, RGT2 controls STD1, And then 3 of these repressors control the low and medium affinity transporters, and the other one, STD1, controls the high affinity ones. So this is some qualitative understanding of the network. But we'd like to do better. Oh, sorry, and I should just point out before we do that, that hxt5, which is an odd one, if we do it, doesn't seem to be regulated by the network at all, or very lightly. So its behavior hardly changes with all the deletions. Okay, so we have that sort of qualitative understanding, and we want to try and do better than that. So the way that we do that is we want to switch from batch culture, where a lot is going on. So the cells are consuming the glucose, but they're also making ethanol, and other nutrients in the media are changing. And so instead, we're going to move to using microfluidics, where we could control the extracellular environment much more precisely. And also the cells, and most importantly, the cells themselves don't control it by consuming the nutrients. So the way this works is, you just saw, I'll try and play it again, is we have these traps. So these things here are pillars that go up from a glass cover slip to a ceiling of a microfluidic device. So they're made of a polymer PDMS. And the pillars are placed just so that they trap the cells. So we sort of flow the cells in a bit like a pinball machine and get them caught. And when they're caught, they happily grow there. And you can see that the buds form and then disappear. No, not anymore. One more time. The buds form and then they disappear, and that's because they're swept away in the flow, which is going in this direction, from left to right across the device. And this allows us to image them for long times. So if we didn't do this, the yeast grow in 3 dimensions, and it's very difficult to follow individual cells because they pile on top of each other. All right, so with this setup, then what we wanted to do is we wanted to try and see more precisely when these transporters come on. So we exposed the cells to rising and falling glucose. And the original plan was to make these very gradually increasing and very gradually decreasing. But at the time, our control of our microfluidics was that the fastest we could really do it reliably was about 30 minutes. So this time scale where they change, a bit hard to see on this plot, but is about 30 minutes or so. Anyway, it goes up to high glucose. We leave them there for about 8 hours. And to orient you, yeast dividing glucose, at least in high glucose, in about 2 hours. So this is 3 or 4 divisions, and then we bring it back down again. With the idea then that we're passing through a range of glucose concentrations, and we should see what these transporters do. So here's an example of experiment. We have fluorescence at the level of the transporter, the GFP tagged, versus time. Glucose, as I said, is ramping up, constant, and then falling. And these are the low affinity transporters, which should be expressed in high glucose. They sort of do what we expect. So there's 2 lines here because just to show you 2 replicates. And you can see that there's no expression, we move into glucose, they start to express, looks like they're going up to peak, and then when we take glucose away, they're removed again, their levels fall. And so this is perhaps what you might expect. When we look at the high affinity transporters, it was a bit more surprising. So it's a bit complicated here because we initially grew the cells in galactose, and these transporters are expressed in galactose. We have some low expression at the beginning, but this gradually starts to fall. When we switch into glucose, there's no expression, and glucose rises up, and these transporters fall further and quicker. And then when glucose is taken away, then we get very high expression. And then the medium affinity ones also do what's very unexpected things. So one of them responds a bit like the low affinity transporters, the red ones I showed at the beginning, but then responds even higher when we remove glucose. Whereas this one completely ignores rising glucose, but responds sharply to falling glucose. So this is much more complicated than we were anticipating based on the idea that it should be an optimal transporter for particular concentrations. So the optimal transporter is being expressed, but other ones are being expressed too. So if I summarize all that, then you can see everything here. What I didn't point out is this hxt5, which is the odd one. So that seems to be expressed in the absence of glucose. And as soon as glucose is present, it's switched off, and it's expressed again. But you can see that the response to an increase in glucose is relatively simple. You have a few transporters come on, whereas the response to falling glucose is much more complicated. Okay, so this wasn't quite what we were expecting, but we can still go and say, okay, this is a lot of quantitative data. We know what the concentration of glucose is in this case, which we didn't know when we were doing the batch experiments. So we can go back to this network. We can construct a mathematical model of it and ask, is everything that's present here enough to describe the data that we're seeing? So just to go through this again, to emphasize a slightly different part of it. So the simplest, so what's key here is that there, as I mentioned before, that there's 4 repressors that are controlling the levels of our output hxtg. And what happens you can think of in no glucose, or very low glucose, these 2 sensors aren't active, which means there's no regulation of, or downgrading of the activity of these 2 repressors. And so they're in the nucleus and they're active in the absence of glucose. And when I add glucose, it's sensed by these 2 proteins. This causes this particular repressor to be degraded. And this one does something different. It leaves the nucleus and aggregates into these kind of foci on the vacuole. But the result is it's not in the nucleus. So in glucose, these both go away from the nucleus. This one, these 2 work the opposite way. Okay, so that in the absence of glucose, SNF1 is active and represses MIG1, and MIG1 is because of that repression is forced out of the nucleus through a phosphorylation. It sits here, and there's no MIG2 expressed because it's repressed by these 2 transcription factors. When I add glucose, the opposite happens. Now SNF1 is inhibited, so MIG1 is no longer inhibited, and it moves into the nucleus. In the presence of glucose, these both are inactive, which allows this one to be expressed. So these 2 express. So what I want, it's complicated, but what I want you the picture you should have is that in no glucose, I have control through these 2 repressors. And then in the presence of glucose, that control shifts in some way to control through these 2 repressors. So I have this sort of interchange of regulation as glucose shifts. Okay, so to do these microfluidics experiments takes a lot of time and effort. So we decided we have to focus on one particular transporter. So we focused on hxt4 because in our initial experiments, it had this very asymmetric behavior where it completely ignored an upshift. So glucose is blocked here, but responds strongly to a downshift. And if we change the concentration of glucose into which the cells are moving, then the response is sort of counterintuitive. So you can see that if we put the cells in lower glucose, then we actually get higher expression of this transporter and a smaller spike compared to high glucose, where there's no expression and a very strong spike when the glucose is gone. So we have this. So then so a few control experiments first. So the spike that we see in the 1% glucose, that's this orange behavior with the orange glucose, is not because of a delay, you might imagine. So that if we make the time in glucose, or in this case, make the time in glucose shorter. So, sorry, before we had 8 hours, and we have a spike here, but if we make it shorter, we also see a spike. Okay, so it doesn't appear to be because of timing, the amount of time in glucose, which by coincidence we picked in a certain way. And it's also generated mostly through transcriptional regulation. So here we have the promoter of hxt4 driving just GFP, whereas here we have the protein tagged with GFP. And you can see the behavior is very similar, except at late times where there's some active degradation of the transporter, which doesn't occur to the GFP. But the spike of expression is the same. So most of the regulation is coming through transcription. So again, this is a good candidate to try and understand. So we repeated the same kind of analysis. We do the microfluidics experiments with mutants where we've deleted different components. So the same 4 as before, but also now including MIG1, the repressor control through here. MIG1 and MIG2 tend to behave very similarly, with MIG1 dominating. So we thought if we understood MIG1, we would be able to understand MIG2, which is true, we think. Okay, so we do that, and then Luis, my student, did all these experiments, and you can see that we have the wild type behavior here, and each deletion gives very different results. So here we delete the 2 sensors, Okay, if I delete this sensor, I get lots more purple. It's more purple than wild type. Whereas I delete this sensor, I get less purple than wild type. Here are the deletion of the 3 repressors, this one, and MIG1. And noticeably, these 2 repressors give very high expression. So you see that the y-axis here has changed from being 4 to being 12, both these cases. So there's these 2 are the dominant repressors that are really keeping levels low. Particularly if we delete MIG1, you get very enormous expression compared to the wild type. Okay, so that was the data that we have. As I said, we can formulate this network into a model, and there's 2. I don't expect you to follow these equations, just for those people who do look at these things, to show you that there's nothing unusual in how we're doing it. So we have kind of Hill function, type control of gene expression, and then we have some regulated degradation as well of the protein through glucose. But it's all a very standard formulation of this kind of network, the translation of it into mathematics. All right, so then given that, we, in collaboration with Vahid Shahrazai at Imperial and Mark Sturrock in Dublin, we fit the model to the data to see if it would describe what we see. And actually, the fit turns out to be pretty good. So again, so the top here are the data, and the simulations are shown below. And you can see that for the wild type, we sort of get everything right. Maybe a little bit too high expression in 1% here, but generally it's okay. If you compare with deletion of the sensors, it's not too bad. And we get these very high expression when we delete these 2, MTH1 and these 2 repressors, this one and that one. Okay, so everything seems to be good. And it seems that in general, the result of this is that, yes, this network can indeed describe this asymmetric behavior where under some glucose conditions, I get no response to an up shift in glucose, but a strong response to a downshift. And we went through, again, quite a lot of effort to ask, can we generate more simple models that also describe this behavior? So maybe some of these interactions are superfluous, but that doesn't seem to be the case. We couldn't find anything where there was convincing evidence that making the model simpler did better than using a full model. Although now that we have the model, we can understand a little bit better where this spike type behavior comes from. So to give you some intuition for that, so I thought of things here. So here's what we're trying to predict, this level of hxt4. Shading again is glucose, and we've got time here. Okay, so in low glucose, we get some expression, then a small spike, and then high glucose, almost no expression, and then a strong spike. And then down here is sort of it's a measure of the activity of the transcription factors. So when we run our model, we're able to estimate the KD of the transcription factors binding to the DNA. And so what I'm plotting here is the sort of the relevant variable, which is the transcription factor concentration against the KD, and then taking the log of that so you can see everything. And what this means then is that when this variable is positive, it means the transcription factor concentration is sufficiently high compared to the KD that it's likely to bind the DNA. And when this variable is negative, it means it doesn't bind the DNA. So up here in the shaded region, we have binding, and without shading, we don't. All right, so given that setup, then we can look to see how the transcription factors, their activity is changing over time and also with different concentrations of glucose. So in this region where I have no expression, you can see I have MTH1 is very active, STD1 is also active, and I have no MIG1 and MIG2, they're out of the nucleus. Okay, when we add glucose, then we get MTH1 is inactivated, in this case it's degraded, and starts to fall, and MIG1 becomes activated and moves in. So then I have some level of repression, which is not very high, because I'm able to produce some of my protein. And then when I remove glucose, then I get a very fast response from MIG1 and a slower response from MTH1. So MTH1 has to be resynthesized because it was degraded, and so it is remade slowly. And this, you can see, gives a small window in here where there's very little repression. And this gives rise to our spike. And in this case, MIG2 never reaches levels where it can control the expression of the hxt. If we go into higher glucose, that changes a little bit, but not very much. So again, MTH1 in this region is dominant. When we move into glucose, MTH1 is degraded, MIG1 moves into the nucleus and starts to repress. But in this case, glucose is so high that I also get significant synthesis of MIG2, and it also actively represses. And the combination now of MIG1 and MIG2 then brings the level of transporter quite low. And when we remove glucose, we get a very fast response from the MIGs, and there's been more degradation of MGH1, so it takes longer for it to resynthesize, and so I get a much bigger spike. So it's really this difference in timing between how these pairs of transcription factors act that allows this spiking behavior. So we get very fast responses from MIG1 and MIG2 when glucose shifts, but slower responses when for MGH1 and STD1. And that allows the asymmetric behavior that we see in rising and falling glucose. Okay, so we have all that. So that's an understanding then of how things are working for this hxt4, the one that we picked. But what we'd like to do really is to understand, well, how are all of them behaving? And we have already some prior information from the batch culture experiments, these ones that I explained before, where we expect that the low affinity hxts are regulated just by STD1, and also, which I didn't show it, the other one, MTH1. And the other transporters are regulated by this combination of repressors. So to see if that is consistent with this behavior that we saw here, this dynamic responses, what we did is we took the model of the signal transduction that we had inferred from hxt4, but then changed the particular hxt that model of signal transduction is regulated. Okay, so instead of regulating the hxt4 promoter, we let it regulate hxt1, and then we use this pink data to try and infer what the values of the parameters should be, of those regulation parameters should be for hxt1. And in fact, the way we did that, we did that much more systematically. And so for each promoter, so if we start with hxt1, we considered all these different models. Let me explain what they are. So the purple model is one where all of the repressors are transcriptionally active. So the promoter is regulated by all four of them. The green model, the promoter is regulated just by the first three, these three. This one is just regulated by this two, that one, and so on. So we consider all different possible combinations of regulation, which gives 16 models, because we have our 4 transcription factors. And then what Mark and Vahid did is they have this approximate Bayesian computation scheme, which they use to fit all these models. And what we get back at the end then is a posterior probability for each of the models based on the data that we gave. Okay, so just to remind you, so for the hxt1, it would just be this particular response here. And then you can see what we get back. That for hxt1 at least, we have one model is dominant, which is this yellow one, which only has MTH and STD1 controlling, providing regulation, so binding the DNA, which is which is consistent with the batch culture data. hxt2 has a pink and blue, so pink has MTH1 and MIG1, which is what we were hoping. For this one, we're not expecting any control from STD1. Okay, in both these models, the pink and blue, no control for STD1. Again, consistent with the batch culture data. And so on, you can go through hxt6 and 7 is also blue and pink, as is hx. Both of them are more blue and pink, which again implies no control through STD1, which is what we saw in the batch culture data. And the only one that's a little bit different is hxt3, which has which one of these models also have this one at light, which has some control through MIG2. So hxt3 should be mostly STD1 controlled, which I think if you look, all of these models are that one, this one, and blue one, and that one. Okay, but also potentially through MIG1. So in general, through doing this systematic model comparison, we get back results from our microfluidics data, which are consistent with the inferences we can get from the plate meter data. So we think we have a reasonably comprehensive understanding of how this network's working. We also, of course, get parameters back when we fit these different models. So we pick the best model and look at the parameters for that model. And we see that there's the two types of repressors work a bit differently. So MTH1, it binds very strongly, these are organized in terms of their affinity. So these are the medium affinity ones. MTH1 binds to them strongly and weakly to the high affinity ones, whereas MIG1 and MIG2 bind strongly to the high affinity ones and weakly to the medium affinity ones. In other words, their regulation is flipped. So I'm trying to show that, we tried to show that here in this diagram, to show that MTH1 binds weakly to the high affinity hxt7 and more strongly to the medium affinity, and MIG1 does the opposite. It binds strongly to the high affinity and weakly to the medium affinity. STD1, which is the one that we think should just control the low affinity transporters, indeed does that, and the higher the Km for the transporter, the stronger STD1 binds. So it really seems to be the dominant control for the low affinity fast transport in high glucose. Right, so then I can put all that together and try and summarize. So that we have our network, which I described, and what this network seems to generate is, in response to an increasing concentration of glucose, is this kind of activity from the repressors. So MTH1 falls first as glucose increases, its activity drops because it's degraded. STD1 also falls, but at higher concentration of glucose than MTH1, whereas MIG1 has the opposite type of behavior, MIG1 and MIG2, where their activity is highest when glucose is highest and falls as glucose decreases. So we have these two opposing types of repressors. One pair is best at low glucose and falls as glucose gets higher, and the other pair is most active at high glucose and falls in the other direction. So given this setup, then the way the promoters are constructed is quite intuitive to try and ensure that you get the transporters put in the right places. So these are the high affinity transporters, which should be expressed in low glucose. And they do that by being being more weakly bound by MTH1. So when MTH1 starts to degrade, they come on. And they're strongly bound by MIG, so that in these higher glucose concentrations, they're repressed. So they only come on in this region. The medium affinity ones are given here, and they have this behavior reversed. So they're sensitive, very sensitive to MTH1, so they're switched off here. But they're less sensitive to MIG1, so they can come on here, but they'll be switched off eventually when MIG gets high. And then the high affinity ones are given up here, and the simplest is hxt1, which is controlled mostly entirely almost, at least in high glucose, by STD1, and so only comes on when glucose is high. So we have this pattern, so we have a network which sets up these opposing behaviors of the transporters, which we're trying to call push-pull, so the sense that as glucose rises, it pushes the repression through the MIGs and pulls repression through the MTH1 and STD1, with the opposite behavior as glucose falls. And then given these different patterns of expression, the promoters are set up with their affinities to ensure that their respective transporters are expressed in ranges of glucose where their transport should be close to optimal. Okay, and this is very similar, this was similar to our eyes, to what happens in Drosophila development. So I saw there was several talks on development before, so you may have seen this already. So there the problem is it's analogous in the sense that you want to express certain genes, the gap genes, which are the ones at the bottom here, in certain positions in space. So whereas in our case we want to express certain transporters in certain concentrations of glucose. And the way that the cell does that is it creates two spatial gradients of regulators. So bicoid, let's focus on that one. Bicoid falls from the one end of the embryo to the other, and nanos falls in the opposite way to the embryo. And this is mimicking, we think, this type of behavior. And then given this spatial structure of the regulators, the promoter structures have evolved to be able to be only expressed with certain concentrations of these regulators so that they're expressed in certain spatial regions. Again, this is analogous to our promoter structures having certain forms, so they're only expressed in some particular region of glucose concentration. Okay, so we think this may then be a general way of solving this problem of trying to match a collection of genes to different values of an input variable, whether it's glucose concentration or whether it's space. Okay, but just to finish quickly, so why have so many? I mean, why not have 3 or 4 hxts? The answer is we don't really know, or I suppose. But what you can see is that what budding yeast is very good at, it's very good at importing glucose. So this is some data from from this paper in 2013. And what they did is they took 46 species of yeast and they compared their growth in 2% glucose, which is 110 millimolar, so high glucose. And what I've done is I took the data now, we took the data and we plotted it where I'm plotting the characteristic of another species compared to the characteristic of budding yeast and taking the log 2 of it. That means that in this region, budding yeast is better than the other species, and in this region it's worse. And you can see that for the yield, so the amount of cells that are produced, budding yeast doesn't do particularly well. There's species that do much better. And also in terms of the growth rate, they don't grow particularly quickly. All these species grow faster than they do. But what they're very good at, it seems to be, is importing glucose. So there's only one species that's better, and that's another Saccharomyces species, which happens to have one extra glucose transporter. So what we think is happening is that there's so many hxts have evolved in yeast is because they specialize to this very fast import of glucose. Now you notice because the yield is not very good compared to other species, they don't use this glucose very well. Okay, so they bring in the glucose and they ferment it, getting only 2 ATP per glucose molecules, whereas they could get much more if they chose to respire it. But by having this very fast import, what we think is they're doing is that they're simply taking glucose away from other competing cells. And so one way to win is simply just to grab all the glucose, and even if you don't use it very well yourself, it stops competitors from using it. And so we think one reason why, potentially why you might have so many hxts is really to try and win this competitive battle for consuming glucose quicker than anyone else. And we don't really understand why we see this complex dynamics. Okay, so as I said, from the argument of that one transporter being better than others, we expect really one transporter to be expressed at a time. The dynamics, as I said before, is much more complex when glucose falls. And of course, when glucose is falling, that's when competition is most fierce. So perhaps then cells are willing to put lots of resources into making sure they get whatever glucose is available by expressing multiple transporters with affinities to make sure they get the right one. I'm not sure. You can also see in batch culture when glucose gets very low, so the OD is plateauing, so it's almost running out of glucose here. You get very high expression of these low affinity transporters to try and presumably get most of the glucose that's left. Also, of course, when you see this variability, you can choose to interpret it in terms of bet hedging, where potentially cells, even in this high glucose, use to express this medium affinity transporter in case glucose falls, so that they're willing to take a reduction in fitness in the current conditions for a possible improvement in fitness in the future. Glucose tends to fall. I don't know. And then I noticed that Gilles Chauvin is going to speak in a few days' time, and he has this very nice, again, Microfluidics setup where they're able to follow cells for very long periods of time. And this is just to show. So this is a measure of cytoplasmic pH, which you can think of as the healthiness of cells. And they see if they grow them in glucose that most of the cells are able to grow for a long time. So they consume the glucose and then they switch to start to consume the ethanol that's produced and grow on that for many hours. But about 10% of the cells fail to do this. So when the glucose runs out, they're unable to switch to use the ethanol and they die. So there's this red line with it getting more and more unhealthy. And so it's possible, it looks like this transition from fermentation to respiration, which they need to consume ethanol, is very costly. And so perhaps you can imagine should be delayed as much as possible. And so again, this high expression here maybe to try and enable fermentation to keep happening in the hope that perhaps glucose will return. So that the switch to respiration never needs to happen. Okay, so I'm almost done. So to finish, I argue that we have multiple of these transporters because of the rate affinity trade-off. And then given that we have that trade-off, then the cell needs to be able to match glucose transporters to the current level of glucose. And we does this through this, what we're calling a push-pull system of repressors, where I have one set that responds to glucose in one way and another set that responds in the opposite way. And then different transporters redecide in different ways so that they can be expressed appropriately. And then finally, but of course, the network does more than that. I mean, it seems like, as we said, multiple transporters are expressed at the same time, but there's some other more complex information processing happening. But just from a methodological perspective, having this time series data is very important But you really see a lot by having changing inputs and following how cells move over time or respond over time. Okay, so then to finish, so this, as I mentioned, this is work. Nearly all the experiments were done by Luis, my former student. Ish did some experiments and helped build the strains. Kevin did the phylogenetic analysis, and all the modeling Luis did with Mark and Vahid, who were in Dublin and London. Thanks to sponsors, and we have a preliminary work is on the bioRxiv too, if you're interested. Okay, so thanks for your attention.
Thanks a lot, Peter. So we have time for maybe a few questions.
Any question?
Yeah, so we're right after the lunch, so everybody is maybe not completely up. So maybe what I suggest is that then we stop here for the coffee break, and we thanks again, Peter. And then we will meet then in half an hour at 3:30 for the next talk of Pascal Hassen.
And please, the students who are going to present the short talk at the end of this afternoon, either send an email with the slides or bring it to me so I can watch them, please.
Yeah, thanks a lot again, Peter.